The first session is called Developing Next Generation Replacement Methods to Model and Study the Brain. And we have our first speaker, Stéphane Jamain. He has completed his PhD at the Pasteur Institut in Paris, where he identified the first functional mutations in idiopathic autism in the neuroligin genes. And from 2005 on, he became a full-time researcher at INSERM, where he developed as well his own research group that was focusing on the genetic and molecular biological aspects of bipolar disorders and schizophrenia. And in the future, in 2026, next year, he will lead the translational neuropsychiatry team that explores neuropsychiatric disorders based on iPS cell-derived organoids. So we are really happy that you can present here, Stéphane. The virtual stage is yours.
Thank you very much. So thank you very much for the kind invitation. I would like to thank the French and the Swiss 3R centers for these kind of invitations. And also I would like to thank Alexandra just before for the very nice introduction and the inspiring work that she did with the team of Thomas and Richard, of course. So now in my team, we try basically to do the same thing, but based on different neuropsychiatric disorders. And the idea is to use induced pluripotent stem cells and to derive them into neurons or brain organoids in order to study the pathophysiological impact of mutations that we identify in neuropsychiatric disorders. So I'd like first to start with remembering how important are the mental disorders in the world. They are among the leading cause of burden in the world. They affect more than 970 million of patients around the world, and it's the first cause of disability-adjusted life years. But they are very difficult to phenotypes, very complex. And as an evidence of this, you can see here the number of edition of the Manual of Diagnostics that has been published by the American Psychiatry Association since 1889, when they started to classify psychiatric disorders. It is still complex, and even today there is no clear definition of the disorders. And just as an example, I wanted to show you that since schizophrenia and bipolar disorder has been split in two different diseases in 1889 by Emil Kraepelin, you can see that many other subgroups of disease have been created along the years. And the more the years increase, the higher the number of groups exist. Now there is some people even thinking that there is a kind of continuum of symptoms between schizophrenia and bipolar disorders. And actually this continuum is something that we can see and between all psychiatric disorders. That is shown here. Because most of the symptoms that exist in all these disorders are shared by many other psychiatric disorders. This is one of the major issues that exist for psychiatric disorders, and this is one of the reasons why it's so difficult to model in animals, for instance, and to study in other models than humans. The other problem is that the definition of psychiatric disorder is mainly based on how the world is perceived by the patients. And it is based only on subjective criteria that are transcribed by the patients to the clinicians, which means that it's very difficult and there is no biological markers of such disorders. So this is also a reason why it's so difficult to model in outside humans. But what we know is that there is a strong genetic contribution to most of the psychiatric disorders. Here, just the example of schizophrenia. And we can divide genetic variants in two major categories. Those that are, here is the frequency of the genetic variants. Here is shown the impact of the genetic variants on the disease. And there is a lot of very common genetic variants that exist in the general population that are more frequent in patients than in controls. And all these variants have been identified through genome-wide association studies on very large cohorts. But these genetic variants are unable to predict if people will develop or not a disease. In contrast, there is very few rare variants that exist, sometimes only in one patient, sometimes in very few families, with a very high impact on the disease. For instance, you can see that protein truncating mutation in GRIA3 multiplied by 50 the risk of developing schizophrenia, or a deletion of 3q29 region multiplied by 100 the risk of developing schizophrenia. So there is now several genes that have been associated with schizophrenia, the same for bipolar disorders, the same for autism spectrum disorders. The problem is that the same genes, mutation in the same genes have been found either in patients with schizophrenia or in patients with autism spectrum disorder or in patients with intellectual disability. And there is no specificity between the genes and the phenotypes that we observe afterwards in patients. And this is a major problem. It's what we also try to understand in the lab. It means that for the same gene with different mutation, why sometimes we have a specific phenotype and why sometimes we have another phenotype. So to give you specific examples, we screen in the lab multiplex families with bipolar disorder, early onset bipolar disorder, with Elisa Courtois, who did a PhD in the lab. And we identified several genes with damaging variants, which were actually mutation rates in these genes were more frequent in patients with bipolar disorder than in the general population. I just wanted to show you one specific example in the gene which is called SMARC2, that encodes the protein that is also called BAF170. And actually, we can see here that the mutation rate of damaging variant in patients with bipolar disorder is 4 times higher than the mutation rate that we found in the general population. Interestingly, patients with mutation in this gene have had more manic, more mania in their disease as compared to the general population of patients with bipolar disorder, which is quite unexpected in the general population. And I wanted to show you one specific mutation that we are currently studying in the lab that actually have been identified through a very large family that you can see here, and which you have been able to see the segregation only in the nucleus part of the family. But this amino acid change, a very conserved amino acid in the protein, and we expect to have a strong phenotype on this with this mutation. So this gene, the SMARC2 gene, actually encodes, as I told you, protein, a subunit that is called BAF170, that belongs to a very large complex of 15 proteins that is called the SWI-SNF complex. This is a chromatin remodeling complex that is involved in many processes of neural development, including gliogenesis, neural migration, specification of neural subtypes, synaptogenesis, cognition, specification of the nervous system, neural stem cell proliferation, and neurogenesis. And what is interesting is that most of these subunits has been associated with several neuropsychiatric disorders, including Coffin-Siris syndrome, which is an intellectual disability, but also autism spectrum disorder or schizophrenia. And now we have mutations in patients with bipolar disorders. The idea is to try to understand why some subunits have, as a consequence, a very severe mental retardation, and some others have psychiatric disorder like schizophrenia or bipolar disorder. To take the example of SMARC2, you can see here all the mutations that have been reported in patients with Coffin-Siris syndromes, in which we also added the mutations that we found in patients with bipolar disorder with the red arrows. So the idea is to try to see what are the consequences of a mutation, sometimes in the same domain, and why sometimes there is an impairment of the brain with intellectual disability, and sometimes there is bipolar disorder. And what has been shown for mutation in this gene in patients with Coffin-Siris syndromes is that for some of them, we had, using brain imaging, they showed either hypoplasia, dysplasia, or agenesis of the corpus callosum that showed the importance of this protein in the brain development. So it is quite difficult to study the impact of mutation in this protein, in BAF170 protein, because the absence of the protein is lethal for the mouse embryos. And the team of Tuok and colleagues a few years ago generated a conditional knockout to see the impact of the absence of BAF170 in the cortical region of the brain. And you can see here that in absence of BAF170 in the cortical region, there is an increase of neuronal cells that are shown here, either here with TBR2 or TUJ1, two specific markers of neurons. And so we can see here when they quantified at different stages of development, the number of neural cells, that there was an increase of the cells in absence of BAF170. In contrast, when they overexpressed BAF170, they had no impact on the PAX6 expression, which is a marker of progenitor cells, but they had a decrease in neuronal cells with, again, TBR2 and TUJ1 used as specific markers. So the model they proposed was that the SWI/SNF complex, including BAF170, was an inhibitor of some specific late progenitor genes involved in the development of cortical of the cortex. And in absence of BAF170, there is no inhibition anymore, and there is an increase of this of the expression of, for instance, TBR2 and the development of the cortex. And in contrast, when there is an overexpression of BAF170, there is an increase of the inhibition and thus a decrease of the cortical size. So studying BAF170 and the different partners, including BAF155, that directly impact or regulate the expression of PAX6, which is known to play a role in the proliferation and the neural differentiation, then it is very important to understand what is the impact of the mutations that we found in patients with bipolar disorder. The problem of studying such development is there is an ethical issue of studying this in mice, as it has been already showed by Alexandra before, but also there is a strong difference in the cortical differentiation between rodents and humans, with more complexity that is observed in humans as compared to rodents. This is the reason why in the lab we decided to try to develop human neurons based on induced pluripotent stem cells, in which we included the different mutations that we observed in patients using the genome edition. We also studied from the keratinocytes from patients that we differentiated into iPS cells with the aim of developing then either neuron or organoid differentiation. And using this technique, the idea is also to be able to rescue the phenotype, correcting the mutation of interest. The result that I'm going to show you today is the first strategy I've been studying from commercial induced pluripotent stem cell, and this is the work that has been developed in the lab by Michele Boniotto. So with William Asaban in the lab, we first started to differentiate the iPS cells either with or without a mutation in SMARCC2 into neurons. And to make a long story short, we've been able to develop neurons and to quantify the number of neurites. And basically, we observe no difference in neurite length and no difference in the number of neurites per cell body between wild type and mutant cells. We also wanted to see the impact of such mutations on the 3D cell culture, and we differentiate our iPS cells into 4 brains using the Lancaster protocol. and at several stages of development, and here it's shown as 60-day in vitro. We've been able to observe the formation of neural rosettes that has been already mentioned by Alexandra before, and there was basically no difference in forebrain differentiations for the earliest stages of development between the wild type and the mutant organoids. However, we try to quantify the different layers at a later stage, and here at 120-day in vitros, we've been able to show a decrease in the number of neural cells positive for SATB2, which is a specific marker for the upper layer in the cortex. So there is a decrease in the SATB2 positive cells in mutant organoids as compared to the wild type organoids. But these are preliminary results unpublished yet, and this slight difference needs to be confirmed, and to be confirmed at later stage with more developed cortical layers in our organoids. But what is important in bipolar disorders is that there is no brain structure abnormalities that is generally reported in our patients. But what is more known is impairment in the connectivity between specific regions. And one of our previous work showed that an impairment specifically between the prefrontal cortex and the amygdala. And the idea would be to study more specifically this kind of connectivity in the context of the mutations that we can find in patients. And as it has been said by Alexandra as well, the huge difference that exists between the human and the rodent brains in terms of the size of the different brain areas and the connectivity between the different areas is so important that it's better to, when possible, to work directly on human cells. So to do so, we try to develop cortical assembloids. I mean, combining, we differentiated our iPS cells into two different spheroids, either cortical spheroids or striatal spheroids, based on the protocol that has been published by Sergei Pasqua a few years ago, with the aim of combining them in an assembloids, corticostriatal assembloids, with the aim to follow exactly what Alexandra showed at the end of our talk. What are the projections from the cortical spheroid to the striatal spheroid, or the migration of interneurons from the striatal spheroid to the cortical spheroid. So we started to do so, and we generated the first assembloids, and we've been able to label specific interneurons here using viral vector, and to measure and to the migration of neurons. And you can see here one example of the one interneuron migrating from the striatal spheroid to the cortical spheroid. And now we are currently measuring and testing the speed and the distance of migration, and to see if there is some difference between mutant and wild type neurons. And I wanted to finish using another gene with other mutations that we generated in the lab in collaboration with the team of Jean-Vianney Barnier, on another gene very important for intellectual disability that is called PAX3, that has been previously associated with X-linked mental retardation, mainly in males, but now many other mutations have been identified even in females. And we wanted with Jean-Vianney more specifically study two different mutations that resulted in very opposite phenotype. One was a severe global developmental delay with microcephaly, generalized non-motor seizures, periventricular white matter abnormalities adjacent to the occipital horns of the lateral ventricles in one males, with a mutation located in the N-terminal part of the protein, and another mutation located in the kinase domain of the protein, with a patient also males with intellectual disability, but with autism spectrum disorder and stereotypic movement no epilepsy, and a macrocephaly. And we wanted to try to see whether our organoid models were able to mimic first, and then to understand why in some times we had microcephaly, and then in other times we had macrocephaly. So we generated corticospheroid for these different mutants, and we differentiated and measures the areas of the spheroids at different stages of development, and we observed, it is quantified here, some difference in the areas of the spheroids in the volume of the spheroids. This has been reproduced several times, and now we are currently quantifying with a large number of, after 3 differentiation, with a large number of spheroids to confirm our results. And finally, we also did we also did imaging, 3D imaging on this brain organoids. Is what you can see here to compare the development of the wild type cortical spheroid with the 2 mutant cortical spheroids. In all cases, what you can see here is the presence of the neural rosette that has been mentioned by Alexandra before. But what is interesting is that, for instance, in this mutant, there is a development in the part, a normal development in the part of the spheroid, but there is one part here you can see where there's no development of neural rosette. And now we try to understand what kind of cells are there, and why there is a difference in the differentiation in this mutant as compared to the other mutant or the wild type one. We also quantify with Celialac and Anderson Bay the volume of the neural rosette for each of the spheroids, and here you can see that there is a significant modification, a significant difference in neural rosette volumes for the 2 mutants as compared to the wild type one. So we again try to understand and go further in the analysis of the kind of cell types that are developed in these organoids to try to understand what are the differences and what are the mechanisms involved in these 2 mutants as compared to wild type. So to summarize, we can say that rodent models are unsuitable to study the human cortical development. Neurons and brain organoids derived from human iPS cells enable the study of a single mutation identified in patients with psychiatric disorder, which is very interesting for us to try to understand the pathophysiological mechanism underlined by such mutations. So neuron and brain organoids derived from the iPS cells are effective tools for studying the early stage of brain development in the context of psychiatric disorders. And the mutations identified in patients with psychiatric or neurodevelopmental disorder, for some of them, impair the cortical development in vitro in our models. So they could be a very good model then to test specific treatment, as Alexandra showed before with autism spectrum disorder, to see if we could propose some specific strategy for some of these mutations. And finally, to conclude, we can say that iPS cells-derived neurons and brain organoids are efficient tools to study the pathophysiological impact of mutations identified in neuropsychiatric disorders. I would like to finish by thanking all the people who contributed to this work, mainly those involved that are currently in the lab and actively involved in the work that I showed today, but many other collaborators as well. I'd like also to thank our fundings from the FC3R and of course the patients and their families, and we'll be happy to answer questions if there is some.
Thank you very much, Stéphane. We are a little bit behind the time, but yeah, I see that you managed.
Sorry
well.
about that.
No problem. So we have maybe time for one short question, and just a reminder, you can write your questions in the chat, and it would be very nice, Stéphane, if you could still answer questions in the chat afterwards, if when
Sure.
the next speaker is talking. So one question, please.
Yes, thank you. Thank you, Stéphane, for your nice talk. You know both worlds, the animal experiments and the IPSC-derived experiments. What do you see as advantages when you use IPSC, which you cannot investigate with animal experiments?
First, in terms of ethical issue, it's much easier to study the IPSC experiment just because you don't have to sacrifice. I mean, what I showed you for what has been done on SMARC-C2 in mice, you need to study them very early in the development between E12 and E15, so, which means even before the babies are born. So in terms of ethical issue, there is a huge difference between the two. But the major other advantage is because now we are able to study them with a genetic background, which is a human genetic background. And we know that the genetic background is very important on the way to develop the brains. And if we can start directly from the patient cells, it would be even better because it would also include all the genes with common factors, I mean common genetic variants that exist in humans and that also can contribute to the brain development.
Thank you.
Okay, thank you very much, Stéphane, again,
My
for
pleasure.
this nice presentation. Thank you.